And it's just. Okay. Well, thank you everyone for attending. My father, Dr. Jack Wands, MD, physician scientist, for the Festschrift and life celebration of his life and his career. My name is Greg Wands, Gregory Dixon Wands, and I'm his son, and I'm also a scientist as well. And I really want to thank you all for coming to this event. And I also want to thank Rolf Carlson for organizing this, and what a distinct pleasure it is to be the one to make the opening remarks and the opening up this session. So, as I mentioned, I am a scientist as well, and Rolf had asked me, do you have any stories or anecdotes that would kind of depict his positive influence on your career? And our careers have overlapped quite a bit over the years. He's actually not that much older than I am. He's about a little over 20 years older than I am. So we've had a lot of time to experience science together. And one of the most memorable experiences I had was actually quite early on when I was in elementary school. So he did his residency at Johns Hopkins Hospital, and he trained as an internist. And I remember that experience very well. And that's sort of where I learned how to speak. And I had kind of a heavy Baltimorean accent at the time. And so when he had his first job offer here in the Boston area at Mass General Hospital, you know, we moved into the Newton area and the Newton school system. And I would have been in 4th grade at the time. And fortunately for Newton, they were very heavy in the sciences at the time. So they had some science projects in 4th grade when I was there. And they were really sort of kit-based. We would get these kits and you'd open it up and there'd be instructions, and you can design and present an experiment. And since I was sort of the new kid, I got my kit and there was a lot of missing pieces in it, and it wasn't really clear on how to do this. And so I remember reaching out to my father because I felt like I was a bit of a pickle, because I was required to go up in front of the class and kind of perform an experiment. And so I was in a bit of distress, and then I came home to my father, showed him my kit, and he's like, this is crap. You know, you can't really get anything out of here. It's like, let me think this one through. I bet I can come up with something a little bit better than that. So the next day, he opened up his briefcase and he opened it up, and he had 3 small Pyrex beakers and 2 containers of chemicals. So they turned out to be copper sulfate and ferric chloride. And they also grabbed some of the table salt off the table. He goes, you know what we're going to do? We're going to grow some crystals. I'm going to show you how to make a supersaturated solution. And this is going to absolutely amaze you and your classmates. So he took those beakers, grabbed a spoon right out of the kitchen drawer, and poured some water on it, and then put the beakers right on the stove top, just an electric stove top. And then he just starts pouring the chemicals in, stirring, pouring, and he just had this big grin on his face. It's like, oh, this is going to be good. Pour it, spit it. He goes, that's it, super saturated. So he goes, your job is to go find some string. So I went and found some string, and he cut some pieces off, and he took those super saturated solutions, all 3 of them. So we had salt, copper sulfate, and ferric chloride. And we put the strings in there, and to me, it just looked like Kool-Aid. It's like there was nothing special about it. But the next morning, when we came back, we were both very excited about it. He said, go ahead, pull the strings out and see what you can see. So I pulled it out, and they were just dripping with these most beautiful crystals. And When I took out the one that was just the salt, I saw diamonds. And then when I looked at the copper sulfate, I saw sapphires. And then when I looked at the ferric chloride, I saw rubies, and I was just astonished. And so we took these strings out and we put them on the paper towel. When we dried them out, he goes, okay, go take those to your class and see what happens. And on top of that, he asked me, do you have any other scientific equipment in there? And I said, yeah, we got some like little cheap kids light microscopes. He goes, let me tell you about electron microscopes. He was really excited about microscopes at that time. So he did the diagram, and he explained very clearly why electron microscope would go down several orders of magnitude in magnification. So between making crystals and the microscope lesson, the electron microscope, we covered a lot of science. We did thermodynamics, we had chemistry, we had wave particle theory. I knew photons, electrons, and all that. And I'm in 4th grade, and I felt absolutely empowered to go into class and just show them my experiment. And that's exactly what I did. So I went up to the classroom, I went to the blackboard, and I diagrammed how an electron microscope works. And I could just see everyone's eyes just get big, because I was able to make them understand it, because my father just had that ability to really take complex ideas and simplify them and give them context so you can understand it. And that empowered me to teach my class how crystals were made and how electron microscopes work. So as I passed around the crystals, it became this I was astonished how excited, and there was this buzz about science going on. And some of the other kids on their own initiative took out the cheap light microscopes, and they were grinding up the crystals and mixing them together and looking under them. And they were just absolutely beautiful. And my teacher at the time, Mrs. Davis, became very excited too, because all these students were saying, Mrs. Davis, come over here. Look at what I have. Mrs. Davis, come here. And I'm just kind of like looking at all this excitement that was being generated based on what my father had taught me. And I distinctly remember my teacher, Mrs. Davis, running by me going, oh, Greg, this is just so exciting. You're definitely getting a gold star. I'm like, a gold star. That's like the highest. Yeah. Everywhere. And so sort of being the new kid, because I talked funny and I wasn't making a lot of friends, but my stature really changed. All of a sudden, I had gotten a lot of respect, and I also made a few extra friends. So that's sort of the experience that my father kind of got me interested in science. And I've been really interested in science. I find everything about science interesting. And that's what I really learned from my father. So with that, I really appreciate you all coming here and celebrating my father's life and his wonderful career. And with that, I will turn it over to Rolf. Yeah. Yeah. Yeah.
Thank you, Greg. And thank you all for coming. I call him. Ray, I just had just a few overview things, just to let you know. You've already seen we have those rooms there. We'll be having the meals. We got the so breakfast, lunch, and dinner. I want to thank particularly the family for Jack, who sort of helped put this together and is helping to sort of like settle the score with the hotel when it ever comes around. Again, thank you all very, very much for coming and agreeing to participate here. I think the response that I got from people was really, really wonderful. And we have people that I know from way back that have come all the way from France, from Turkey, from other parts of the country. And I think it's a testament to how great Jack was. People have asked me since he died, many people have asked me sort of like, can you tell us a little something? What were the special moments? What was it like? How was it? What you must have shared a lot of things with him. And truth to be told, it had gone on so long that a lot of it was like, I got to think about this, because we were together all the time, and the reality was, this is Jack, whether he was 40 years ago or whether he was just shortly before he passed away. It was just Jack. But then I decided to go back and think about some of the stuff that we did together, and I wanted to try to think, like, how would I characterize my experience with Jack? And that would be the 45-year adventure with Jack. Because it really was, that was the year I decided to wear my lederhosen, I think, to the party, the Christmas party that we had. So yeah, I decided that was probably the best way to describe it, because we did so many things together. And I thought when I really thought back on, like, said, did we really do that? And a lot of it stems from what Greg had said, his enthusiasm towards science, his enthusiasm towards getting things done, trying things new, and you know, just coming up with this idea. And at the time I would think, like, what? But then we would just do it. And some of the stuff was really, I'll tell you a few things. I'm to this day, I think, like, I don't believe we did that. But the science was certainly the most important thing. And it was all about the liver. And in the course of the many years that I worked with them, I worked on every one of those things. And you probably wonder, like, if it's all about the liver, why does it say neurodegeneration? Well, because that was one of those weird things that sort of happened, that brought in a whole new way of looking at certain things, particularly Alzheimer's disease. And that stemmed from one of the first interesting stories regarding the next things that we did, which is Jack was very quickly to embrace models and try to do things when new stuff came around. And then when monoclonal antibody technology hit, I basically became an antibody factory, because anytime someone mentioned the proteins, like, we're going to make an antibody to that. Ralph will make an antibody to that. Let's make an antibody to that. So Dr. Jerry Gross at the time had an interesting story with him. Was he had published a paper, I think in 1948, on the structure of elastin, and he described certain thread proteins in the structure of the protein that turned out to not be part of the protein. What it was actually a byproduct of the trypsin preparation he used to digest the protein. So he actually had to retract the paper, or retract that portion of the paper describing the thread protein. But Jack was, we'll make an antibody to that. So I did. I made lots of antibodies to it. And then we did some work with them, and then Dr. Gross did some work with them. And then he went and tested every single type of tissue, human tissue you can think of. And the only thing this was a pancreatic protein. The only thing it cross-reacted with was something in the brains of Alzheimer's disease patients, which was, talk about serendipity. So that brought in a whole new sort of angle, including the work with subsequently the work with Dr. Delamonte, that actually brought another whole thing too. So it wasn't, in the end, it wasn't all about the liver, but a lot of it was. In terms of models, this is one of my favorite stories. Like, we were doing a lot of cell culture work. You know, we grew all sorts of cell culture lines, a lot of hepatomas, and he wanted to have his own hepatoma line. So again, he came up with this idea, and when he just approached me and told me, it says, they're going to be bringing you a sample of a hepatoma right from the patient. I'm like, okay, great, we'll do that. We'll do whatever you have to do to it. And what I wasn't expecting was the sample was quite a large sample. Like, he walked into the cell culture room with, had to be like half the lobe of a human liver, with what I thought was cauliflower growing in it. And he goes, well, I'm like, I guess that's the tumor, right? He's like, yeah, that's the tumor. So we did, actually, with the help of one of the first associations with Chinese scholars back in the 1980s, we actually did manage to grow a cell line that came to be known as Focus, or Friendship of China-US. And it's gone on to be a lot of interesting work that subsequently came based upon that cell line. When you're looking for tumor antigens, you basically nowadays you just sort of like find out what's the protein you're looking for and whatever and stuff like that, and then you start to work with that protein. That's pretty typical now. But back then, Jack didn't want to approach it that way. So I was like, well, we want to make a tumor antigen, see what we can do. So why don't we just immunize them with the cells? So we took these focus cells and injected them into the mouse. And somehow the mouse survived because I was literally injecting 2 million human tumor cells into the vein of a mouse. Needless to say, the spleen you need to take out to basically make the monoclonal was enormous, because it just had all those things. But then it became like a fishing expedition, which is how, okay, I got all these antibodies, what are they responding to? So that's where some of the work that was done, particularly by students back then, started characterizing all these things that came to be associated with tumors and hepatoma, including one of the proteins that proved out to be very, very important, ASPH, or aspartyl beta hydroxylase, which apparently is which is very involved with the metastasis and progress of human cancers. And that all, again, came upon because Jack decided he wanted to just go for it, just do something like that. When we work on hepatitis B, we made lots of hepatitis monoclonals. We had a peculiar trait back then, like to be able to easily identify the antibodies, he decided to give the mice all names. So the first one was Harry. Harry didn't really bring us much in the way of antibodies, but Harry too was a gold mine. They brought a lot of things, including IgMs, IgGs. It ran the gamut. And now, what can we do with this? Well, diagnostics was a big thing that'll come in a little later. You can take these things. At the time, it was developing assays to test for the various proteins. And so he thought that we could go ahead and use those. And that led to a collaboration that we'll talk about later with Dr. Bellet. But he also wanted to cure hepatitis. So we needed a lot of those antibodies, like a lot. So I bought the biggest column I could possibly find in the Pharmacia catalog or anywhere, short of getting something industrial size. We still have that column. It's sitting in the lab in a corner because I just somehow can't get rid of it. And the reason I had to buy it was because we were using an IgM, and you had to do that by just separation chromatography. You just like run it through the column. And I'm thinking like, well, this column's gigantic. It's obviously it's going to take forever for that antibody. No. Fluid dynamics. You have something that much with all that fluid. When I opened up that column, it was like a fire hose. It's just like whoa, like the stuff. Everything's just started coming out, and I said, like, if I'm going to purify this antibody, I better slow it down. So we did, and we actually managed to isolate 2 grams of an IgM, a monoclonal IgM to hepatitis B. Well, what are we going to do with that? What do you have to treat? You can't treat a human with that because you can't. So he decided we would just go to Alamogordo and treat a chimpanzee. So we found this chimpanzee, Fred, that was a chronic carrier of hepatitis. And we injected him with 2 grams of a monoclonal IgM. And for 4 days, Fred had no detectable hepatitis B surface antigen. But then, because it was a mouse-based antibody, Fred started to make his own antibodies against it, and that sort of like ended that. So we never actually didn't cure. We just gave him a few free days where he probably wasn't quite so infected. Fred didn't last much longer. He was already an older chimpanzee, and once again, Jack decided we could probably do something with the liver. So they sent me the entire liver, and it was in the freezer for years until I finally decided I don't think I'm going to be using Fred's liver anymore. So that was some of the fun science stuff that we did. But also the very, very important thing was the progress and the growth. I lost something. Yeah, the progress and the growth. I didn't imagine when I started as a technician that like 10 years out, Mass General was going to float $1 billion bond issue so that they could basically buy the building 149 in Charlestown. And that was kind of like being a kid in the candy store, because Jack was like, we gotta build a new lab. I'm like, okay, great. Well, how are we going to pay for it? So just go to these meetings. So we went to these meetings that they were holding, and they were like, well, how much money can I have? Well, just tell us what you want and we'll get it. And they did. So we built the molecular hepatology lab with just all this money from Mass General Hospital. And I got to buy everything. It was awesome. Ultra centrifuges, you name it, whatever we needed that we didn't have. Most at that time in the lab, we mostly had the small stuff because we had to leave all the big stuff behind. But it was a spending spree. It was awesome. I don't think I ever spent that much money in my life until we built the Liver Research Center, because then it was the same thing all over again with even more money. So in terms of growth, we grew from 1, 2, 3, maybe 4. We have Russell Brown. He came here. He was a technician with me way back in the early 1980s. We started with a small group and ultimately ended up a very, very large group over the course of the many, many years. It's gotten smaller now, but that was one of the most challenging things and one of the most fun things too, was actually being able to build those 2 laboratories as Jack became more and more known. In terms of the growth, the other important thing was, to a certain extent, I didn't have to travel the world because the world came to us. I was thinking about it, and we have had research associates, research fellows, people on sabbatical, students from all continents, with the exception of Antarctica. And we had a very, very large contingent over the years from Japan. Dr. Sasaki's here to talk about that. We had a very fruitful collaboration with France. Dr. Bellet is here, and Dr. Puisier was here. Through Hubert Bloom in Freiburg, we had a very, very productive association with German universities, and from everywhere else up subsequently, most recently with China. So that was one of the really, really fruitful and rewarding parts of actually working with Jack, was all that. Meeting all those people and all these lasting friendships that will continue for many years to come. And as I said, it's the people. The people also very, very important. The ones that came from all around the world, and also the ones that he tutored, mentored here in the United States, particularly in the clinical fields. Once he became director of the Division of Gastroenterology for Brown, that's something that he took particularly great concern for, was that everybody got an absolutely quality grade A education on the clinical side, as well as continuing on with the research. And then, of course, there was the fun. Up on the left there, the upper left, you can see is actually one of the earliest cake hour pictures from 1996. And then it was actually started with the German contingent that we had at the time. And there's a couple of them right there. There's Jasper and Martin. And then Jack found I started playing the piano for the lab, which started off on a whim. The GI lab at Mass General Hospital used to have a Christmas party, but it was at the director's house, and it was a dinner where you went, you ate, and you left. And he didn't invite the technicians, or anybody subsequent below the fellows. So this crazy guy that I work with, I don't know if you knew Jeff Week or not, but this crazy guy that I worked with decided that we should have a Christmas party for everybody else. So he knew I played the piano, so we had to hunt down a piano. So we found one at the adult psychiatric unit at Mass General Hospital. We found a piano. So they allowed us to go and take it, but we didn't listen to them, and they told us not to go through that door. So immediately we set off the alarm. But in the process of getting it there, I just kept playing as we were going through the elevators and across the hallways, and it ended up everybody in the hospital thought that was great. Mass General was much smaller then. But that started us having these Christmas parties. First in the GI lab, then we went to the cancer center, where we had them all the time, and I'm still doing that. As a matter of fact, I'm going back on the 14th of December to play for the Christmas party at the cancer center. And then Jack made an association with Sylvia Mubayed at the Cav restaurant, which was just getting started in Providence, Rhode Island. And then we just took over their restaurant for a night. Whenever we had these Christmas parties, we'd get a piano. And I watched people like Dr. Kelly's children grow from here to now. They think they're in this first year, second year of college. The same thing with Dr. Promrat. He's not here yet, but his kids started here. Now they're graduating college. So those were some of the really, really nice, fine parties that we had. And then as an excuse to bring everyone together, he had these dinners. We started these dinners at the AASLD, which many, many people, anyone coming to the AASLD is invited to attend. We've gone up as much as 40, 50 people who had previously been with the labs who are coming back. So this is something that was very, very important to him. And if you're wondering why you're here at the Four Seasons, there's a reason for that too. I decided I wanted to pay off some school loans rather quickly. So I lived 2 blocks away and I saw this building going up. And I was like, what's that? Turns out it was the Four Seasons Hotel. And then I was reading the paper today, one day at lunchtime. I said, well, you know, I was a bartender. Maybe I could be a bartender. So I sort of read in the paper that they were interviewing 2 blocks away from Mass General. I said, okay, I'll just go to an interview. I didn't show up dressed or anything like that. I was wearing a t-shirt and a sweater. And they wanted me to go to 2nd and 3rd interviews. I said, why? I said, like, well, because we think you'd be a great concierge. I'm like, what's that? And they explained to me what it was. They realized I was giving tours of Boston. I knew all this stuff and history and stuff like that. I said, okay, let's give it a try. So they hired me part-time. After a couple of years, I think Jack saw an opportunity. So it suddenly became like, well, what do you think? Maybe I could stay at a Four Seasons? I'm like, yeah, probably. And so he did. He stayed at a lot of them. But we also, in return, the hotel was very, very good and accommodating, and would allow us to bring rooms here. As I got to know people, they would like give me a little extra break so I could make it competitive to be in this hotel during the ASLD instead of at the Marriott. And then we had the dinners here and stuff like that. So there was a good association with not just this Four Seasons Hotels, but in his case, Four Seasons Hotels all around the world. So I thought it was an appropriate place to bring it all back to, because this is somewhere where, you know, sort of he enjoyed being, where we all enjoyed being, and we had many, many good, successful dinners and stuff like that. But for everything that took place, the most important part was the man. You know, I lucked out. I was really, really lucky. It was one very quick interview. He was thrilled. He was so excited about the work that we're going to do. My other option was to basically, at the time, the first 2 offers I got was, one was to sort of grind up large amounts of calf thymus to isolate stuff. And I just got done grinding up during UMass large amounts of human placentas. And I was like, I don't want to grind anything up anymore. So I went, and plus because of his enthusiasm, I went with him, and it turned to, you know, such an enriching experience, such a wonderful experience. The legacy that he left with me is one of having had a professional life that was, as I said, beyond compare, and also gave me the opportunity to meet all of you people and be associated with you people, to keep science going, and to, you know, sort of really thrive in life in so many, many ways. And so and as you all know, we all were affected by him. We all were influenced by him. And he was just always Jack. For 45 years, he was Jack. I didn't really notice any changes until I went back and started looking at the pictures. And I was like, yeah, oh yeah, we were younger then. That's for sure. So there was a next, as I said, we're more than happy to have Dominique here, Dominique Bellet. He was involved with Jack in production of diagnostics for, and he'll talk about that, for proteins and stuff like that. It was funny back then, this was around the time that the AIDS virus was being discovered. And there was this battle between the US and the France as to who really discovered the AIDS virus and stuff like that. So there was, you know, scientific conflict, or whatever you want to call it. But the Boston Phoenix found, a way in which sort of the French and the Americans could get along and cooperate together. So they published this article, Double Takes, or When Two Heads Are Better Than One. And you notice here in the pictures here, we have Jack and Dr. Bellet, people were getting together, working together at the time. I think this is when you were putting together the radioimmunoassay for alpha fetoprotein. And they did this article about all these successful pairs, and I don't know how they got wind of the 2 of you, but they included this in the article. And you notice this nice little thing here. Even if we speak a special language, the general recipe for collaboration is trust, Dr. Bellet says, in some very important projects. there's the possibility that investigators may keep data for themselves. Dr. Wong concurs only by sharing data can you make research go. The danger of collaboration across the oceans that one group can be so focused on the glory of it, and they can keep the data for themselves. I don't know if that was a dig at either side of the ocean because of the HIV thing, but they certainly they were recognized for their collaboration by the Boston Phoenix, which isn't published anymore, but I did find this online. Just there. So as I said, it was with great pleasure to have Dominique come and speak and give us his reminiscences and sort of experiences with Jack. Thank you all.
Hello. Thank you, Rolf, for your invitation. Indeed. Oh, thank you. Indeed, 40 years ago, a long time ago, it was the expertise of Jack's in immunoassay development that led to our meeting. I would like to meet with Jack, and I went to Boston at the GI unit, but unfortunately, he was in Philadelphia at that time for a meeting on liver diseases, I think. So hopefully in his secretary offices, you have pictures of all the members of the GI units, including obviously Jack. So I look at the pictures, I look at Jack, and I went to Philadelphia at the meeting, yes, trying to find Jack. But it was really easy because he has his Victor Hugo beard. And at that time, it was quite unique because it wasn't really on fashion. Today, it's more in fashion, that would be more difficult to find Jack. But at that time, it was very easy. So I find Jack. And I was really impressed by his eyes sparkling with intelligence. I was impressed by his radiance, by his kindness. And as we said, it was love at first sight between us. We do appreciate the Jack and I, so we decide to work together. So we went with my wife in Boston from France for a sabbatical year, and we work with Jack in Jack's lab in the GI unit managed by Kurt Iselbecker. It's a nice lab of Jack. And at that time, I meet, hopefully, Rolf. Rolf is really the pillar of his laboratory. Yes. And as Jack, Rolf became my friend and a deep friend as for Jack. So at that time and during, I guess, 40 years, you have a family atmosphere in Jack's lab. And even during the journal club, you have a family atmosphere. Also because my wife, that you can see him, was doing some delicious cake to pass on my broken English. As you know, I still have a broken English, but thanks to the cake of my wife, at that time, everything goes well. And with Jack, and under his supervision, was developed an assay for the alpha fetoprotein. Here is this assay, was highly specific for the detection of liver cancer. Yes. And if you look at, for example, these pictures, you can see that really this assay was wonderful for the detection of liver cancer. So the discovery of this assay by Jack make the headlines and the front page, for example, of the New York Times, and the headlines in every journals, ever in United States, ever in France. You have the front page, yes, of the New York Times. You have the front page of many French journals, including Le Monde, for example. Everywhere you have some articles about this essay. And as shown by Rolf, please. No. Yes, Rolf showed this article in the Phoenix, who celebrated the alliance between the French and the American for doing science together. And this alliance continue with the coming in France of some very good research fellows, including, for example, Mehmet Öztürk. We came from my lab in France to work with Jack. And Mehmet and Jack has published a very important paper. This paper, indeed, described for the first time a link between a food element and liver diseases. And this link was through a mutation in the p53 gene. And at that time, Mehmet doesn't probably remember that, but at that time, this article was considered as one of the most important articles of the year, if not the most important particle. So congratulations for Jack and Mehmet. Mehmet, after that, has many of the research fellow Jack made a brilliant career in Turkish, in France. He's now professor, he's still professor, 40 years after that, he's still professor. You have another very good research fellow, Alain Puisieux. We came from Paris today. Thank you, Alain, for being here there. He's quite a little bit sick. It's okay? It's okay, thank you. Good. But he comes from Paris, like Mehmet. He published some very good article with Jack too. This one, for example. And he became also very successful. He is now the scientific director of Institut Curie. Institut Curie is probably the best renowned institution in France. He's a scientific director. Congratulations, Alain. Anyway, with Jack and with Mehmet, we continue to develop various immunoassays for the detection of various cancer. We have published, for example, this paper about the use of immunoassays for the detection of human chorionic gonadotropin. Anyway, we have published a lot of papers with Jack and Mehmet for the various assays, and interestingly, most of these assays are still in use 40 years later. So anyway, once time, I guess it was at the time where we had the headlines, for our work on immunoassays for AFP with the front page of the New York Times and so and so. Jack, tell me, Dominique, you know that you have the 5,000-year man, Jesus, Mahomet. You have the 500 years man, Mozart, Chopin. You have the 50 years man, Churchill, De Gaulle. And you have the 5 minutes man, you and me. But really well remember that. But I guess that Jack is already and at least a 50 years man. And you, Jack, my master and my friend, we are not about to forget you.
Well, the assay that he mentioned that got all that attention in the various newspapers also got attention on television. Notice that this scientist.
The world's deadliest form. There has been a major advancement in the detection and the potential treatment for it. Here's ABC's George Trader. This scientist is performing the first blood test ever developed. Substances that can specifically identify the cancer protein in time for doctors to treat the disease. We are quite optimistic that this test may be used as a screening procedure for the early detection of this usually fatal tumor in high-risk populations. Liver cancer is almost always fatal, but 98% of people contracting it in this country. The annual death toll was 10,000. Worldwide, almost 1 million people a year die from it. In fact, liver cancer is the #1 cancer killer in the world. Liver cancer is most common in Asia and Africa because that's where hepatitis is a public health problem. And 95% of liver cancer victims had hepatitis or have been exposed to the hepatitis virus. And we want association of the treatment will be available worldwide within a year. It will be crucial for the millions who come down with hepatitis every year, and those at high risk for developing this disease, like healthcare workers and male homosexuals. A male who is applying carrying the virus would have perhaps as much as 50% chance of developing a tumor, and a female 20% chance. I believe that this solid tumor in time is curable surgery. But the real significance here is that a lot of oil antibody has been made to detect this specific cancer, raising the hope that other molecules can be made to detect other cancers. George Strader, ABC News Follow.
Those mice in that video had nothing to do with it. Nothing. The cameraman just wanted something that would look cool. That's all. Well, what about these mice here? I'm like, oh yeah, that's really cool. Yeah, well, this was all before the law, the Animal Welfare Act. Because there's no way I would have had those mice with tumors that big in the lab. But they were just like thrilled. They thought that was the greatest thing ever. So there are a number of people that wanted to say something about Jack that couldn't be here. Also, this has happened when he passed. So I'm going to include some of those today for the presentations. The first one is from Professor Shinji Tanaka, who was a research associate with us at MGH Charlestown, who was very, very productive. He was the one that did some of the transfections of the cells to produce and overproduce ASPH and various other proteins associated with the carcinogenesis process. So this is his message when Jack left us.
Hello everyone, I'm Shinji Tanaka, Professor, Tokyo Medical University. Today, we gather to remember and honor our beloved creator and collaborator, Jack Raymond Wallace. We are deeply saddened by his sudden passing due to heart disease. From 1994 to 1996, I had the privilege of being a postdoc researcher at Master under Jack's guidance, and our collaboration continued to this day. Together, we published over 20 research papers, building a testament to his passion for, and science we have healthy. Jack was not only an efficient scientist, but also a warm-hearted and caring person. He touched our lives profoundly and supported us like a true friend. As we grieve the loss of this remarkable individual, let us cherish the memories we shared and carry on his legacy by continuing our research in his honor. Our thoughts go out to Jack's family during this difficult time. Rest in eternal peace, dear Jack. Your light will continue to shine in your hearts. Thank you.
He's Dr. Tanaka was actually going to come to the ASLD, but then when he found out that Jack had passed, he canceled, but then we scheduled this, so we went with the video. As you know, Jack was very influential in the hepatitis B virus, in studying the hepatitis B virus, which surprisingly was, I think, discovered in our lifetime, or my lifetime. One of the things that we did was, after we made the monoclonal antibodies, he showed through the use of immunoassays, radioimmunoassays at the time, because we hadn't quite have the enzyme immunoassays yet, so we're talking lots of radioactivity. He wanted to show that the hepatitis B virus was not the unique, single, unchangeable virus that everyone thought it was at the time. And the way that he did that was collect samples from around the globe. We are talking lots of samples. We got them from the Gambia. We got them from Aborigines in Australia. We got them from South Africa. We got them from Europe. And what the technician at the time, Nancy Brown, ran lots and lots and lots and lots of assays. And then through an actual sort of mathematical analysis of the results, he showed through this thing that they call signature analysis that, in fact, the hepatitis B virus was not the same thing in various parts of the world, that there were indeed variants and there were indeed different subtypes of the virus, as it were. His interest in hepatitis obviously brought to the laboratory two people who have been some of the most productive people that we've had. They've been with us over 30 years, I think 30 years. They are the world-renowned experts in the molecular hepatology of the hepatitis B virus. When I try to describe it, people in sort of layman's terms, I say, like, well, they knock one of this nucleotides out of the virus and it spins this way instead of spins that way. That's sort of an idea of, like, that's how intricate the work that they do is. So Dr. Xuping Tong and Dr. Jisoo Lee have been sort of stalwart colleagues for quite a long time, brought a lot of recognition and acclaim to the laboratory. And it's my pleasure to introduce Xuping here to come and give us a talk on the whole hepatitis story that he's been working on for, what, your whole life?
Thank you very much, Bobby. Thanks very much for the kind introduction, and it's also okay. It's really challenging to talk after Dr. Dominique Bellet. Je parlais aussi un petit peu français before, because Jisoo and I, my wife and I, we worked as PhD candidate in INSEM Unit 271 under Dr. Christian Teppel. So that's the history. And also in the pictures that Rolf showed, about Jack's, former trainees. And also I saw the face of Dr. Hubert Bloom from Germany, from Freiburg. And actually my first stop outside of China was Freiburg. I stayed there for 1 year and sequenced the clone of duck hepatitis B virus. So really I learned molecular biology from Dr. Hubert Bloom. So I want to thank him. So, to forward, okay. So basically, let me see. Yeah, so Jisoo and I came to Boston at MGH Cancer Center in 1993. And we worked as postdoctoral fellow with him, with Dr. Jack Wans, until 1998. And afterwards, we moved with him here to Rhode Island to continue our work. So Dr. Wans is a really very kind, gentle mentor. He always is encouraging, you know. He is always very positive, you know. So basically, what today I'm going to talk about, the title is called Searching for the Keys, because you may try to find the keys, but probably you never find the keys. Depends whether you are lucky or not lucky. So that's why I give this phrase. So we are really looking for the receptor for the hepatitis B virus, which really Dr. Wans was very much interested. But we primarily work on the duck virus. So what is a receptor for an enveloped virus? Because hepatitis B virus is an enveloped DNA virus. So a receptor is a cell surface protein that can bind to the viral envelope protein. And that mediated initial step of viral entry into the cells, which is attachment. Attachment followed by entry. And viral receptors could be a single receptor, but for many viruses, there are multiple receptors. There are a sequential binding to different receptors. And such receptors may determine the tissue or species specificity of infection. In this regard, we know very well that HBV only infects humans and chimpanzees. So it's very much host specific. And also, as you learn from the name, you know, hepatitis B virus, so it's really targeting the liver. So it's also tissue specific. So in this regard, a receptor may determine that, although subsequent events may also determine the specificity. So after the virus binds to its receptor, then because it's an enveloped virus, so the envelope has to fuse with the cellular membrane, which could be on cell surface, or after the virus binding, the entire virus is engulfed, called endocytosis. And after endocytosis, it goes to the endosome, and the pH changes. And with the pH change, or sometimes with proteolytic cleavage of the viral envelope protein, then the viral membrane would fuse with the endosomal membrane, and eventually the internal cargo, which is the viral capsid, would be released, and the viral genome would be released, and eventually would start viral replication cycle. So in terms of HBV, so as I said, HBV is actually the prototype of the hepatotropic DNA virus. So we call hepatinovirus, okay? And after HBV is discovered, people found the related viruses which infect ducks, woodchucks, ground squirrels. So for the duck virus, it's called the duck hepatitis B virus, and shortened as DHBV. So after the discovery of these related viruses, the DHBV has been especially useful because it's a more simple than HBV. It's a simpler virus. And also because ducks are very cheap. You can get ducks and infect ducks with the DHBV. Or you can get their primary duck hepatocytes. They are also susceptible to the DHBV infection. So this is a very good animal model. So lots of essential steps in the life cycle of this family of virus was obtained from studies using DHBV as the model system. So before we came to Jack's lab, as I said, you know, Jisoo and I were working in Lyon, France with Dr. Christian Tepo. And what I did was actually, we were among the first few labs which found the pre-core mutant of HBV. Pre-core mutant means that the virus have a mutation in the pre-core region, which would specifically prevent the expression of viral E antigen or HBeAg, okay? And before that, in the Mediterranean region, you know, people know that usually if a person lost the HBeAg and becomes anti-HBe positive, usually the disease would get better, you know, you know, but somehow in the Mediterranean region, some people who are anti-HBe positive still have severe liver disease, still have very high viremia. So they call that probably they are infected with the variant, Mediterranean variant. Several groups, including Howard Thomas group from London, and Bonino's group from Italy and Dr. Christian Teppel's lab independently found that the so-called Mediterranean variant actually is a specific viral mutant. It has a mutation which would prevent the expression of E antigen. So therefore, the virus is E antigen negative. So I was, because I learned the sequencing technique from Dr. Bloom, so I was able to sequence such a mutant. And we also got the first genome, and we showed that such a mutant is still replication competent. It's just like wild-type virus. It's not defective. And furthermore, Jisoo found that that mutation is prevalent in the genotype D in France, but not genotype A in France, because we know that in Europe, the major HBV genotypes are A and D. So the Mediterranean variant actually corresponds to genotype D, while genotype A would rarely develop that because of the constraint of a base pairing requirement of something, So anyway, but Jisoo also did something with Dr. Lucina Kova, which she worked on the duck virus. Because we know that if we study HBV, we know that the HBV produces tons of so-called surface antigen, HBsAg. And after cloning of the HBV genome and the DHBV genome, people located the coding capacity for that surface antigen. But people also found that the surface gene coding sequence is preceded by another sequence, which is also open, which also has the coding capacity. And that region people call pre-S, because it's pre-surface, okay, so it's a pre-S. But what's the function of pre-S? Nobody knew. So Jisoo did something which is introduced a mutation into a restriction site in the pre-S, and found that when she injected such mutant into ducklings, some ducks were able to get the virus replicate. And afterwards, she found that such DHB, when she sequenced again, found there's a mutation to restore the open reading frame. So anyway, she did some work with the duck virus. So now we came to Jack, we said, we did lots of work on HBV mutants. It's so interesting. Let's continue work on the DHB. Jack said, yeah, that's a very good idea. But, in terms of important things in the hepatitis B virus and the liver cancer, Jack said that we probably still have 3 major issues. One, how does the virus, hepatitis B virus, get into the liver? That's the receptor. What is the receptor? Nobody knew, okay? Then he said that, the importance of the human immune response. Can we, cure HBV infection, And certainly the other thing is just like Rolf talking about, liver cancer, How does HBV cause liver cancer? Nobody knew. So he said that those are the important questions, So then we thought, okay, maybe we can work on the HBV receptor. But since it's so difficult, because in cell culture, there is at that time, no system of cell culture which can be infected with the human virus. So we said that let's take one step back. Maybe can we first work on the duck virus? After we clone the duck DHBV receptor, then maybe we can look at the human virus. Jack said, okay, very good. So that's how we started with our postdoctoral work on the DHBV receptor. So here is a diagram showing that whether for HBV or DHBV, as I said, the surface gene coding sequence is preceded by the pre-S. And actually, pre-S also has the ATG codon, can start the translation. So you can start translation either from the S gene ATG and produce a protein, which is a major amiable protein, which we call the small amiable protein. Or you can have you can start from the pre-S region. So therefore, the protein would have both pre-S and S domain. And that's called the large amiable protein. Okay, that's for the DHBV. So the DHBV has a small amiable protein or large amiable protein, with the larger one has the extra pre-S domain absent in the S protein. In the HBV, the story is more complicated because you also have a middle one. You have a small, middle, large. So the small has the S domain alone, middle has the pre-S2 plus S, while the large has pre-S1, pre-S2, S, okay? And the previous work on the DHBV suggests that the receptor probably binds to the pre-S domain rather than the S domain. So we started by generating a fusion protein between GST and the DHBV pre-S domain. And in this way, we found that a duck liver protein about 170 kD can bind to the PS region. And that binding seems to be physiologically relevant, because if we add a lot of duck serum into that, then the binding to the GSD-PS fusion protein is reduced, suggesting that the duck virus, the real virus, can compete with the artificial GST fusion protein for the binding to this P170 protein. But certainly, as I said, the receptor may control the specificity of tissue tropism of the virus. We look at the P170 across different duct tissues and found that it's found in many different tissues, including those tissues which are not infectable. So that's not a very good sign, okay? But on the other hand, the positive thing was that the binding site, we mapped the binding site by using deletion mutant of the PS, deleting from the N terminus or deleting from the C terminus. And in this way, we can narrow down the binding site to that region of the PS, say 87 to 102. That region is actually where several neutralizing antibodies bind. So that's interesting, because why do neutralizing antibodies block virus infection? Most likely, they prevent the binding of the receptor to the virus. Okay, that's how neutralized antibodies often work. So that's something positive. And eventually we did the peptide sequencing and the cloning, and we found that because a short time, another group, which is Don Gannon's group in UCSF, they found another protein called the GP180. And turned out the P170 we found and GP180 they found were the same protein. And it turned out after cloning, it's the duck carboxypeptidase D. It's a carboxypeptidase. Okay, so it's the same protein. So here is a kind of a more diagram, more visually you can see that, on the left side, we are looking at, this P170 CDN. And once we clone that, we transfect it into a human kidney cell line, Bosc cell line, and you can see, P170 expression. And on the right side, you can see that the virus can bind to these cells. At the very lower bottom, actually, we have a deletion mutant to remove the C terminal, about 80 amino acids. And that DCPD can still be expressed, but it's no longer able to bind to the DHBV. While we were searching for the P170, so we, as I said, we use a deletion mutant to try to locate its binding site. In this way, we found that some deletion mutant can bind to another protein about 120 kilodalton, we call the P120. So can bind, so certain N-terminal deletion mutant and certain C-terminal deletion mutant, and also certain double deletion mutant can bind to this novel protein, P120. And its binding site is even smaller than P170, but it is part of the P170 binding site. So they have the overlapping binding site, okay? And that binding site is also part of the neutralizing epitope, so that's interesting. More importantly, P120 was only detectable in the liver, kidney, and pancreas. Those are the tissues which DHBV can infect. P120 is not expressing in other tissues, so it can explain the tissue tropism of this virus. And certainly, the final thing is to see what is P120. So we did the peptide sequencing and the molecular cloning, and turned out that the P120 is the P protein of the duck glycine decarboxylase, DGD, okay? So here is a diagram. You can see that, we use the N-terminal deletion mutant, C-terminal deletion mutant, or double deletion mutant. On the top, you can see P170 band, So initially, the first, the leftmost lane is the complete pre-S domain. It has a strong band of P170, but no P120. But afterwards, with some deletion mutant, P170 is detectable, P120 also became detectable. And in certain deletion mutants, they only bind to P120, not to P170, okay? So here is a diagram. So before our work, other people have generated monoclonal antibodies, just like Jack did, you know. But Jack did not raise monoclonal antibody to the DHBV, I think, if I'm correct. Okay, yeah. So that's by other labs. So here, the red things are actually the neutralizing antibody binding sites. So these are called the neutralizing epitopes, while the green ones are non-neutralizing antibody binding sites. So you can say that in the middle part, there are clustered neutralizing epitopes. And actually, the P170 binding site overlap with such a clustered neutralizing epitope, while P120 binding site is part of P170 binding site and is also part of the neutralizing epitope. If you look at the below at the sequence, you can see that the P120 binding site, which can be narrowed to a very short sequence, at the very end is actually 2 arginine residue, okay? So this sequence, in principle, you know, if there is a proteolytic cleavage by furin or furin-related endopeptidase, then probably the P120 binding site can be exposed. And that probably, we hypothesize that can switch the virus binding from the initial P170 to P120, and probably P120 mediated the membrane fusion. So it's kind of sequential events eventually lead to the productive infection. So that's our working hypothesis. So actually here is a diagram showing that if we transfect the DCPD into box cells, just I showed you before, and then incubate with DHBV particles, if we say incubate for 4 hours or 16 hours, if we immediately at the 0 time point, immediately afterwards, look for the viral signal, either looking at the viral DNA, which is on top of DHBV DNA, or looking at the viral envelope protein, the L protein, large envelope protein, you can see that initially at time 0, there is a strong signal. But after 2, 4, or 6 days, on the left, the cells, intracellular signals of the virus declined over time. But on the right side, if you look at the culture medium, the viral signal in the culture medium increased from 2, 4 to 6 days post inoculation. So therefore, DHBV binding into these DCPD reconstituted cells, they can bind and probably enter, but eventually they would pump out, get out of the cells. So therefore, it does not lead to productive infection. So we did some experiments suggesting that P120 is probably important for productive DHBV infection. There are several points here. The first point is that it's well known that if you culture the primary duct hepatocytes in vitro for prolonged time, then the cells are getting less and less susceptible to DHBV infection. And if we check for the P120 and the P170 expression, we realize that the P120 expression declined over time, while P170 did not. The second thing is that for those kind of cells, they have been cultured for too long. If you add the P120 cDNA, so restore P120, then we can rescue some of the early steps in the DHBV life cycle, suggesting that it has a function. You put it back, you know, what is lost, you put back, you know. And then the two lower points was using cells, fresh cells, which are susceptible to DHBV infection. Either at that time we had was antisense iron, that's a long time ago. If we add antisense iron to P120 so as to reduce its expression, then we found that the DHBV susceptibility of such cells is reduced. Okay. And also, we can add antibody to P120 because P120, part of P120 is available on cell surface. If you added the antibody can bind to the protein and internalize, and eventually the cellular pool of P120 would be reduced by the antibody treatment. Using this treatment, we found that we can also diminish DHBV infectivity. Here is an example of this kind of experiment. So here on the right side, okay, so either we have no antibody added, or we added the P120 antibody immediately after incubating the primary duct hepatocytes with the DHBV particles. after virus inoculation, if you add this P120 antibody immediately or 18 hours later for some time, and then we can see that we can markedly reduce DHBV infectivity, which is looking at the viral envelope protein, okay? And at the lower part, we are looking at viral DNA. So either looking at the viral envelope protein or looking at the viral replicative DNA, it's reduced by P120 depletion through its antibodies, okay? Since we added the P120 antibody after the inoculation with the cells, with the virus, so the conclusion is that the P120 may work at a post-binding step of the DHBV life cycle. So here is kind of a summary, of these clustered neutralizing epitopes, there is also the P170 binding site and also the P120 binding site, and the P170 subsequent study from us and others suggests that the P170 binding is host specific. On the other hand, as I said, the P120 is only distributed in DHBV infective tissues, okay? So the two together probably can explain the host and tissue specificity of the DHBV infection. But unfortunately, we were never able to reconstitute a productive DHBV infection in cell culture by adding both cDNAs. We did not achieve that. So as for the HBV, actually the neutralizing epitopes are much simpler. There are only 3 neutralizing epitopes in the HBV large envelope protein. So again, as I said, the HBV large envelope protein can be subdivided into 3 domains: pre-S1, pre-S2, and S domain, okay? So some antibodies against the middle of the S domain can neutralize HBV infectivity. And some antibodies against the N-terminal pre-S2 domain are also neutralizing. And furthermore, antibodies against the N-terminus of the pre-S1 are neutralizing. Then, more recently, it was discovered that heparin sulfate proteoglycans, which is a sugar, can bind to HBV and serves as the low affinity HBV receptor. So that explains why anti-S antibodies are neutralizing. More importantly, from Beijing, Wenhui Li's group discovered that NTCP, sodium taurocholate co-transporting polypeptide, is the high affinity HBV receptor, okay, NTCP. And so therefore, the NTCP binding site can be blocked by the anti-PS1 neutralizing antibody, thereby inhibiting HBV infection. What remains unknown today is how does the anti-PS2 antibody neutralizing HBV infectivity. So a few more words about NTCP. I'm getting to the end. So NTCP was fished out by Wenhui Li's group from Beijing as a binding partner for the myristylated pre-S1 peptide 2-48. And also before his work, other researchers have already mapped down the HBV high affinity receptor binding site to the first 48 residues in the pre-S1 domain. So therefore, so other people really prepared him for his final work, because they have narrowed down the receptor to a very small part of the pre-S1 domain. Importantly, very importantly, when he transfected this NTCP cDNA into a human hepatoma cell line, HepG2 cell line, the cell line now becomes susceptible to HBV infection. That is a really strong evidence that this is a receptor, because it can convert something from non-susceptible to susceptible. So that's something missing in HepG2 cells, right? And also NTCP could also explain both the tissue and host specificities of HBV infection. So the final thing I think is unclear, because the NTCP was fished out as a binding protein for the myristylated pre-S1 peptide. So the question is whether NTCP can bind to the full-length L protein. Or maybe NTCP binding requires prior proteolytic cleavage, just as we proposed for P120 for DHBV. That's not clear. So my final slide is that, so in a way, you can say that when we promised the Jack that let's first work on the DHBV and then get to the HBV. We made a big promise which did not realize, because you can see that although DHBV is a very good animal system for the HBV, in terms of the receptors, the DHBV receptors and HBV receptors do not overlap. Okay, they use different molecules as their receptors. So that's something which kind of unfortunate for us, So, but on the other hand, there are some common features for the HBV and the DHBV. That is, the large envelope protein for both HBV and DHBV, they are myristylated. So they are modified by lipid at the N-terminus. The N-terminus would be a glycine because the methionine at the very beginning is cleaved off. So the second residue, which is a glycine, would be the attachment site for this myristic acid. And such myristylation of the L protein is not required for virus production, but is essential for both HBV infectivity and DHBV infectivity. So as I said, Wen Hui Li's group uses this myristylated pre-S1 peptide to fish out NTCP. And it's also interesting that what does NTCP bind? NTCP actually bind to glycine or taurine conjugated by acids, okay, glycine conjugate. So probably that makes a way for the NTCP to bind to the myristylated N-terminus of the L protein. And in terms of P120, what is P120? As I told you before, P120 is the P protein of the duck glycine decarboxylase. So probably somehow P120 can also associate with this myristyl port of the L protein, although we don't know. But that's some kind of speculation as to So working on the receptor, as I said again, it's not that you want to find the key, you do find the key. It's something can be frustrating, can be depends on your luck or whatever. But anyway, we published a paper on this since 1995. And the final paper on this was 2010, so that's a really 15 years of span. So it's a long time. And we thank Jack for his patience, for his encouragement. And, and also, as Rolf said, Jack is really good at making antibody. And also in the pictures, he showed you a German fellow called Jasper zu Putzlitz. And actually, more recently, we have been working on, and we somehow accidentally found a fusion protein between the polymerase protein of HBV and the envelope. So it's a polymerase envelope fusion protein. And for that, we need an antibody to show that it has the part of the polymerase. So we bought an antibody, which is called 2C8 from a company. But eventually, when we looked it up, we found that this 2C8 antibody was published in Journal of Virology several years ago with Jasper zu Putzlitz as the first author. So again, Jack is the expert on monoclonal antibodies. Monoclonal antibodies on everything. Thank you very much.
Spencer, is Goldstein there on? He's the remote presenter. Is he ready? Because I can have him go now rather than. Well, these are two short videos, so we could show those. Let's just have them. We'll be there in like 2 minutes. So go ahead and play the video. The next two. We have two more videos from Dr. Masafumi Shimoda and Takahiro Noda. Also from Professor Chair Dr. Bei Sun.
I'm Masafumi Shimoda. I'm here from Oxford University, Boston. Dear Dr. Hans, this week we heard the news of your passing away. We will remember the fantastic time we had on our family in Providence around 2008 to 2011. You showed me a role that with them and vision. And we are very grateful we got the opportunity to meet you. Thank you so much. We need you. Goodbye. Goodbye.
The director of General Surgery of the First Hospital of Harvey Medical University. That one was honorary professor of our hospital. I believe none of us can deny that Professor Wang was more than willing to revisit Harvey Medical. We shared great and unforgettable moments together, leaving us with a beautiful memory. Spending hours at Harvey Ground Theater and exploring several tiger parks in Heilongjiang province and enjoying delightful meals at a little restaurant. Our academic visit to Xi'an and the college lunchtime completion were equally cherished. Professor Wang was a remarkable teacher. He nurtured young and middle-aged students like Festschrift, who have now risen to become the head of the general surgery, excelling both domestically and internationally. His kindness and thought extend to our students, offering them unparalleled level of tolerance and I distinctly remember a time when, despite recovering from physical trauma, he recently appeared for a regional schedule and engaged with them in academic communication, leaving a deep impact on our path. His outstanding academic achievement and the strength of a character make him a true role model, worthy of everlasting admiration and remembrance.
Okay, just if, and there's plenty of water, coffee, anything like that. If anybody ever wants to, during the course, feel free to take a break. So our next presenter is actually remote. He's Jorge Goldstein. And he's going to talk about something that many of you may or may not know about, but in-ray wands, an issue regarding patents that he helped litigate, right, Jorge? And he's going to offer us his take on all of this. Go for it.
Good morning. Are you going to control the slides? Can you hear me? Ralph?
Hold on. Gotta get sound.
Can you hear me?
Yeah.
And maybe could I have the first slide? Or should I share? Let me share. One second. Oh, good. Okay.
Now on the slideshow.
Can you see the slide?
Yeah, yes. Go to the slideshow.
Great.
Up above.
How's that?
Got it.
Good. Well, thank you very much for inviting me. It's an honor to be here and to tell you a little bit about Jack's life outside the lab and inside the courtrooms. My luck in meeting Jack was that at the time he was doing his fundamental work on IgM monoclonal antibodies at the Mass General Hospital. I was a young and rather inexperienced patent attorney representing the Massachusetts General Hospital. And I'm talking now about the late 70s, early 80s. And the Mass General, and through Jack Wanz's lab, was experimenting with the very first early university corporate relationships in the history of biology. This was really pioneering at the time. And the concept that every major professor would have a startup like it happens today was really not even in the imagination of anybody in the administration of the Mass General or in general at Harvard Medical School or wherever. But Milstein and Kohler had recently invented a way of making monoclonal antibodies through hybridomas at the Medical Research Center in Cambridge, and Jack had immediately adopted the fusion techniques and had started making antibodies. And his work resulted in IgM monoclonal antibodies of high affinity to the hepatitis B surface antigen. And he went to Marvin Guthrie, who was then the head of technology transfer at the Mass General Hospital, and told him the following story. Together with Doctors Zurawski and Shoemaker, Jack, together with some venture capital money, had formed a company called Senecor. And the Zurawski, Shoemaker, and Wanz invention of high affinity IgM monoclonal antibodies against HBsAg was of great interest to this company, Senecor, who wanted to commercialize assays for the hepatitis antigen. And so a deal was done between the Mass General and Senecor, under which Senecor had exclusive licenses to all of the technology coming out in this field out of Jack's lab. Zurawski and Shoemaker had been working with him, and they were named as co-inventors in a patent application that I filed on behalf of the Mass General in 1980. And there was one particularly good hybridoma cell line called the 5D3 line, which in order to